Hello there, it's Mrs. Scoville, and we're on day 2 of mutations. We are in our new unit, genetics. So a quick review here before we get into our objectives for today's lesson. Gene mutations, which we talked about yesterday, those are going to be a change in a nucleotide sequence. So there we're looking at small changes in genes. And remember, there it can be very small. It may only involve a single nucleotide change. The consequences of these, though, sometimes were changes in proteins, which can have consequences for the cell. And this can result from errors in DNA replication, so copying, or in transcription, so making RNA. Chemicals, so their mutagens can cause gene mutations, and actually viruses can cause mutations as well. So in today's lesson, we're going to be focusing on the other type of mutations that can occur, and these are going to be larger chromosomal mutations. So in today's lesson, our 3 goals or objectives are to compare and contrast the severity of gene and chromosomal mutations. Yesterday we talked a lot about kind of the different levels of severity with genes. We'll think about chromosomal today. Identify the origin of mutations in chromosomes. And again, these are going to be larger, and also distinguish between the types of chromosomal mutations. We'll see there are several types. First of all, let's just start with the definition of a chromosomal mutation, chromosome or chromosomal. So here, we're talking about the changes in the actual structure of a chromosome. So yesterday we were talking about genes, which are going to be small portions on a chromosome. Today we're talking about larger, actually chunks of a chromosome that will be missing. And this is usually going to be either losing or gaining. When we're comparing gene and chromosomal mutations, those gene mutations that we learned about yesterday are much more likely to be inherited. This is because the effects of gene mutations are likely to be less severe. As we saw, some gene mutations lead to absolutely no effect in the cell. Chromosomal mutations, on the other hand, are going to be much larger and as a result can have very big effects. Many of these chromosomal mutations we actually never even see because they're going to be so lethal. The embryos will not survive if they occur in meiotic cells. So that would be in our egg and sperm. As a result, we do actually see more disorders that have gene mutations in people because the chromosomal ones do not survive. Chromosomal mutations, we saw yesterday with gene mutations that it was usually an error in DNA replication. That can be the case as well in chromosomal mutations, but this is going to be, again, larger scale. So we'll see a specific example of this. Most of the issues that happen with chromosomal mutations are going to be from chromosomes being where they're not supposed to be. So in mitosis, we can get chromosomal mutations if we don't have proper lining up during metaphase. So this is our normal picture of how chromosomes would line up during metaphase. If instead you had one that was kind of to the side or just not even lined up correctly and the cell didn't catch it, that can actually lead to a mutation down the line. Okay, because the chromosomes are going to be where they're not supposed to be. So in that same line of thinking, if the chromosomes don't separate down an anaphase like they're normally supposed to become individuals, or if you have one or more go the wrong way, this leads to improper numbers of chromosomes, and it can actually lead to a mutation as well. And then finally, this is something you wouldn't necessarily think about in mitosis. The spindle is pulling on these chromosomes, and that exerts a lot of force and pressure, and sometimes the chromosomes actually will break or snap. And again, this is going to be during anaphase when they're pulled apart. A lot of the chromosomal mutations that we think about, though, are going to occur during meiosis, and this is because the ones that occur during meiosis are the only ones that are going to make it into offspring. So just like we saw with gene mutations, that is going to be true as well for chromosomal. So meiosis is going to be the main driving factor for passing on genetic disorders. So there in meiosis, the main culprit of chromosomal mutations is going to be improper crossing over. So as a reminder, crossing over, that's where you have 2 similar or homologous chromosomes that line up and swap alleles. So they're going to swap portions. So a lot of these things we're going to see that is improper. It doesn't get swapped correctly. And also similarly to mitosis, we're going to see a specific instance as well where chromosomes don't go the way they're supposed to, or they don't separate, and again, this is going to be an issue during anaphase. This is going to happen in either the first or second part of meiosis. There are going to be 5 types of chromosomal mutations: deletion, inversion, duplication, and translocation/insertion, as well as nondisjunction. We're going to go through each type now. Alright, the first deletion is just what it sounds like. Something's going to get deleted. This is usually caused by the chromosome actually having a piece that breaks off. And in a deletion, the piece of the chromosome that breaks off is actually lost. So it's no longer a part of the chromosome, it goes somewhere else. So again, just that's what it sounds like. It gets deleted, it breaks off, and it goes away. Alright, the next type, inversion. There, again, the chromosome segment breaks off, but this is kind of funny. It inverts, so that means it flips around backwards, and then it'll actually reattach in the same spot. So that looks like this. So again, it breaks, the chromosome will break, it'll have a point where it breaks, then it gets twisted around, and then reinserted. So again, these breakage points, this could happen during crossing over. It's just going to flip around and it's inverted. When this gets read during transcription, it's backwards, so it's no longer correct. Alright, our next type here is translocation or insertion. And we can look at this first in pictures, then in words. So during crossing over, normally you have 2 homologous chromosomes, and as a reminder, they're homologous because they're similar in usually size, and they also will contain similar genes, which we call alleles. In a normal crossing over situation, that's going to be what is exchanging the portion of the chromosome. In a translocation, if we look over here, we're going to have 2 chromosomes that aren't homologous. They're not similar. They should not be paired, but for some reason they incorrectly pair during prophase. So what will happen in that case? Again, they get together and they swap, but the genes are not similar at all. So you end up with portions that are swapped between 2 very different chromosomes. So I've said that here, it's 2 chromosomes that aren't homologous, they're going to swap. And here we say part of one chromosome is transferred to another. I like to think of them as swapping. That's usually what happens in a translocation. But if it's not reciprocal, so if, for example, part of this chromosome came off and it went into the other one, but nothing came from the other one, then we just say it's an insertion. Okay, so if it's not reciprocal, it's referred to as an insertion, but in both cases, some portion is going to the other one. In a translocation, it's both. Okay, and then finally, a duplication. This is going to be in a DNA replication issue. So that first way that we could end up getting an issue with a chromosomal mutation, and that would be a duplication, where we actually duplicate or get too much of the gene sequence. So again, it's just basically an entire gene. So instead of a few nucleotides being duplicated or added, here it's basically an entire gene, a whole section of DNA. And you can tell that this makes it longer, so that's actually one way to identify a duplication mutation. Alright, our final type here that we're going to discuss is a nondisjunction. And this is a little bit different than the others because this is really just a failure of the chromosomes to actually separate during meiosis. So this was one of the ways that we could get a chromosomal mutation, and this can again occur in either meiosis 1 or 2, and we'll see this in just a second to help visualize this. The problem here is this leads to gametes, so again, egg and sperm, that will have either too many or too few chromosomes. This leads to many genetic disorders, although again, there actually could be many more, but the embryos do not survive. So the most common example is called Down syndrome, which you probably are familiar with. And then 2 others that you may not be familiar with are Turner syndrome and Klinefelter syndrome. Those are instances where there's improper numbers of the X or Y chromosome, and they have some pretty significant defects for the people. Interestingly enough, in Turner syndrome, sometimes the people actually have webbed feet, so it's kind of interesting. So if we look at this nondisjunction, we're going to look at it with Down syndrome here in the example, and I know there's a lot going on this slide, so I'll walk you through it. So Down syndrome, for starters, is called trisomy 21, and it's called trisomy 21 because there are 3 copies of chromosome 21. We have 23 pairs of chromosomes, number 21, and someone with Down syndrome, they have an extra copy. This is going to result from what is called a nondisjunction. In this case, it's meiosis 2, but let's look at it in both cases. So normally in meiosis, we've got kind of a normal situation going on over here. Your homologous chromosomes line up from each other and then become separated. In a nondisjunction, they don't separate from each other like they're supposed to. So in meiosis 1, they both go to one side. What this means is in the next round of cell division in meiosis 2, you have one cell that has basically an entirely extra set of that chromosome. So you should only have one homologous chromosome go here, one go here, but instead they both go in the same cell. At the end of that, you end up with one extra copy of that chromosome. You're only supposed to have two, you end up with three. That is a trisomy. Okay, try for three copies of one chromosome. And the opposite cell in that case, it got gypped, it doesn't have the second chromosome like it's supposed to, so at the end it only has one copy of each. So this would be a monosomy. Okay, so mono just meaning one, tri meaning three. That just refers to the number of copies. On the other case, which is what we see in Down syndrome, in meiosis 1, everything proceeds normally, it's okay. The homologous chromosomes separate from each other. The problem becomes in meiosis 2 that the sister chromatids don't separate into individual chromosomes like they're supposed to. So this is only going to happen in one cell, and it just leads to one gamete or egg and sperm that is going to have a trisomy, where again you have three copies, and one monosomy with one copy of the chromosome. The other gametes that are made will be normal from that initial cell. You can notice here, this meiosis 2, remember, is very similar to mitosis. So this is one of the disorders that we see in mitosis that can cause chromosomal mutations. Okay, so now we're going to go through a short animation just to remind us of these chromosomal mutations. So again, deletion, it's taken away and it's gone. Duplicated, it's copied, this will make the chromosome longer. And again, that's going to be an error in DNA replication. In the inversion, we see the gene come out, flips over, goes back in. Everything's backwards now, it won't be read correctly. Remember, this is a special type of translocation. One part comes off, goes in the other one. The first chromosome gets nothing, it's shorter. Our translocation here, where it's swapping, I'm going to see a portion of each goes on the other. They swapped, they translocated. Alright, so we're going to look at these one last time here in terms of letters. So we used the fat cat yesterday to look at the difference in the gene mutations. We're just going to use the alphabet here to look at it. So if we had an original chromosome that was ABC, little asterisk being our centromere, DEF, in a deletion, again, it's going to be of entire genes or large-scale things. So here, the entire B gene is missing. That would be a deletion. It's missing. It's gone. And notice, again, our chromosome is shorter. Duplication, you duplicate a gene. So if the B gene was duplicated here, it'd be ABBC. And again, now our chromosome is actually longer. In inversion, just what it sounds like. A portion pops out and swaps around. So here it looks like we go A, B, C, D, E, F, and here it's A, E, D, C. So the portion C, D, E, it swapped. Although B and F went there as well. So let's see, it actually looks like B, C, D, E swapped, and we were left with A and F normally. So that whole thing inverted. You can see now if this gene was trying to be read, it would be wrong. And then finally, a translocation. This one's a little bit harder to see because essentially we would have to have two chromosomes. So if we start out with A, B, C, D, E, F, and we had a second chromosome, that was G, H, I, J, K, you can see they both swapped an entire portion. With that, we are at the end of our lesson. We will do a lot more practice with identifying each type of these chromosomal mutations, but do go ahead and write your complete summary.